So you know about the Ediacaran, you know about fossilization. Next on our list is the Cambrian explosion. And so this is probably the single most important event that happened in the history of life. I'm pretty sure it is, actually. It's the first appearance of abundant metazoans all over the globe. Okay, so if we find Ediacaran fossils out there a little bit earlier, they were soft-tissued, simple organisms. This, all of a sudden, we got some pretty complex ones. And the diversity is just out of this world. We're going to see one other major diversity event in the Paleozoic, but it's going to be like filling in the niches that the first explosion didn't do. So here we go with the Cambrian explosion. So the Cambrian explosion, in fact, starts with, well, that layer down here on the left-hand side. Those are called Trepignis pedum beds, if you will, and that is from Newfoundland. And so that forms the base of the Cambrian, in fact. But most people, when we think of the Cambrian, we think about these sort of critters that are in the middle here. That's a trilobite on the left-hand side and a trilobite on the right in the middle. And so that's Phylogeny. And then I think the other one is Paradoxides here. And you've already seen Paradoxides before. And then on the right-hand side is how we know that the explosion even occurred. Because we see these crazy things that show up. And that's really probably some sort of primitive shrimp that's over here on the right-hand side. That comes from the Burgess shale. We're going to talk about the Burgess shale in addition to what it's not just the Cambrian explosion, it's also the strata that these things occur in. And so this is episode number 12. Here we have the Cambrian explosion. We're exploring the evidence for ancient life on this planet. So Eukaryotes show up in the Archean probably, and certainly by the Proterozoic they're around. And you already know about the stromatolites that were around in the Archean and on into the Proterozoic where they really hit their peak. And so those things had like chloroplasts in them. They were able to photosynthesize. And about that same time, the first eukaryotes kind of show up in the Proterozoic. And so what am I really talking about? Probably some of the first algae and plants and things like that. And then also things we call protists. Now, you may recall hearing about somebody named Carl Sagan. Carl Sagan was married to a very famous biologist. Her name was Lynn Margulis. And Lynn Margulis wrote a book that was pretty famous back in the 1980s. It was called The Five Kingdoms. And it was not just plants and animals and fungus. It was also the protists and in the bacteria and archaebacteria, as it turns out later, archaea as we know them today. So we have these 5 kingdoms that suddenly became 7 after a while. And so, in fact, we know that the viruses are outside of that sort of organism, level of classification scheme themselves. They're in things that have realms even, right? So, and they're just as complex and related to one another. They're just as interesting in many ways, I guess you could say. But in fact, okay, we have a little bias being animals, I'm sure. But in fact, we want to see where those animals came from, but they really probably came out of the protists. So the protists are our first step along the way here. Amoebas. Things called foraminifera. Foraminifera leave an interesting fossil record. In fact, they're probably one of the most used fossils for identifying what age sediments are throughout the Cenozoic. So foraminifera are pretty important. They've been important at least since I'd say since the Cambrian probably, because they made shells. And so foraminifera had shells. There's also things called radiolaria. Radiolaria are single-celled organisms as well. Foraminifera, single-celled organisms. Amoebas are obviously, but they don't have a skeleton, so we don't have much of a record of them. But and then also on top of that, we're not going to get very deep into the forams, but forams in and of themselves have a whole bunch of people who study them. And they mostly work for oil companies, and so in deep sea cores and things like that, they can tell exactly what age they're drilling through. based on the forams that are in there. So, oil companies probably financed a lot of paleontology over the years just to figure out what age the rocks they were drilling through. Xenophyophores is the last stop along the way here, and there may be a fossil xenophyophore that we recognize from the Ediacaran actually. So they probably, each one of these things probably went all the way back to the Ediacaran at least. And so the plants, the protists, the fungi, and even the animals probably went back then. So that's our eukaryotes, right? So these things are in the Cambrian. They're around today still, and so they were nearly everywhere. So if we had something that just said all of a sudden there's life on this planet, these are the layers we'd look at. We'd look at the Cambrian. The Ediacaran was kind of hard to find. They didn't even find it until the 1940s, right? And even then, it's pretty rare because we don't get those age rocks exposed everywhere. But Cambrian rocks are pretty much everywhere. They're in Missouri. They're in places like Utah, Nevada, Mexico, all over. Not in the Caribbean so much because they're not old enough rocks, but in the Baltic and Siberia, Russia, Persia, all over. Africa, India, Australia, so everywhere else there are Cambrian age rocks. Gives us a record of the earliest metazoans. That's the thing that we call them, right? The eumetazoans in particular. So if we look at the plants, you're looking at kelp, you're looking at things that are amoeboids, and these are all things you see on the left-hand side of this photo right here. Things that would grow and form part of the eukaryotes, but in fact, so there's a radiolarian down here in the middle and the bottom down here. An amoeba over here on the left-hand side. Euglena, it looks like, over here in the middle on the right-hand side. Maybe some fungi. There's kelp up here in the upper right. That's a plant, right? So it's a sea plant. If we looked at xenophyophores are these single-celled organisms that live in the deep oceans today. There's not much record, but people have interpreted Paleodictyon, this fossil that you see on the right-hand side, from the Ediacaran in the Flinders Range. That's been interpreted as a xenophyophore there. I don't have any photos to show you of a xenophyophore, but they're not really that abundant today, except in the deep sea, on the seafloor. Next, we get into the animals then, right? So the animals are a branch of those eukaryotes, and they're a little bit more sophisticated than what you've already seen with the protists. So what's the next level up from a protist? Most people would agree that it's the phylum Porifera. Porifera are sponges. And among the sponges are some really interesting ones that show up in the Cambrian and then they rapidly disappeared. They're called archaeocyaths. But today they have been classified, they're assigned to the Porifera based on their structure and based on their life habits and their symmetry and so forth. They have radial symmetry, but they are considered to be a stem group of all animals, in fact. And so they're a sister group, in fact, of what we call the eumetazoans. So what's the next stage up from the sponges? That would be the cnidarians. So cnidarians you've already had some experience with in the Ediacaran, things like jellyfishes. And jellyfishes are just one life stage of what a cnidarian may have. Some cnidarians have an alternating life phase of going from a polyp to a medusoid, to a polyp to a medusoid, and back. And so there's all these sort of like complex sort of life behaviors that you get out of cnidarians. You don't see that in the poriferans. The poriferans do have specialized cells, however, and they have a specialized skeleton as well. And that's what makes archaeocyaths so special. Archaeocyaths only lasted from the lower Cambrian to the lower middle Cambrian, and then there are some post-middle Cambrian examples that show up in the late Cambrian. But by the time you get to the Ordovician, gone. Well, maybe. Maybe they show up as another group later on. In fact, they're still sponges, in fact, but they are regarded as what we call sphinctozoan sponges. So sphinctozoan sponges show up like during Silurian and Devonian time. But for now, we're just talking about sponges. So here's some sponges down here that are actually what we call irregular archaeocyaths. They look like sponges, but they're not spicules in them. It's the only difference. It's more skeletal. So it's not something that will break apart into spicules. Here's some more Archaeocyaths from the lower Cambrian here. These things formed reefs. That's why Archaeocyaths are so important. The only reefs that you've seen before this were stromatolitic reefs. So the stromatolites were important in the Proterozoic. When you finally, the Ediacaran was a time after a very rapid and brief cold snap, but you really don't see a whole lot of stromatolites in the Ediacaran, but you do find the Ediacaran fauna, and then by the time you get into Cambrian, all of a sudden, boom, reef makers show up again, and that's our Archaeocyaths here. They're unusual, so they look like a little wheel almost, right? So they have a segmented sort of like partitioning that goes around that wheel. And so you can see that in the right-hand side over here. Some of these have been broken open here as well. So, but in general, they form a sort of cup shape, if you will, that is conical in form. And if you do a cross-section of it, it looks like a series of spokes around a, well, like a wheel, if you will. So here's what they look like in reconstructions for the Archaeocyaths. They break them into two parts. They call them irregular and regular Archaeocyaths. The irregular Archaeocyaths have a much more well-defined partitioning. The irregular Archaeocyaths look much more like sponges, in fact. So these are just a handful of the kinds of Archaeocyaths that you might see in some of the reefs that were in the lower Cambrian rocks around the world. I've seen some of these in Antarctica. In fact, one of the ones that we found in Antarctica was 97 centimeters long. It's like, wow. I mean, that's huge, right? It's almost a meter tall off of a seafloor, or maybe buried in sediment. We don't know exactly how it lived, but it could have been keeping pace with sediment as sediment was around it. But in fact, it was filled partially with sediment that would indicate that it had been excavated at some point and then preserved at this 97 centimeter long Archaeocyath. Amazing sort of creatures. They're animals, okay? So they're not plants now. Here you can look down on some of these Archaeocyaths in a reef. And so this is actually reef rock from, it's a limestone. It's a reefal limestone from the lower Cambrian. That's one of the distinctive things about the lower Cambrian, in fact, is that you get Archaeocyath reefs. Well, where do you find them? Oh, you find them in Nevada, you find them in Antarctica, you find them in Australia, you find them in Siberia, you find them anywhere where there's lower Cambrian rocks exposed pretty much. In Poland, I think there are some. When it comes to sponges, the record's pretty actually sparse in the lower and middle Cambrian. Well, not so much the middle Cambrian, but the lower Cambrian for sure, because they just hadn't developed the sclerites yet, or these spicules that would actually hold the whole skeleton together. And so here you have an example of what it looks like to be a sponge here. That is a sponge on the left-hand side. There's one on the right-hand side that's been kind of squished in a shale here in this case. So, yeah, sponges and Archaeocyaths are pretty much like this. Most people recognize today. Among the other true metazoans, this is outside of that group of sponges now. We had other things, and that would be the cnidarians, and then a whole group, and the cnidarians we refer to as the radiata because at least one phase of their lifestyle are the medusoids. The medusoids would in fact have radial symmetry, and everything else on this planet has bilateral symmetry for the most part. There are things that have six-sided symmetry, five-sided. Most of them you can divide by two, however. And so that would be pretty much everything, including us. We have bilateral symmetry as well. And so we're part of the bilateria as opposed to the radiata. So that's the eumetazoans, are this group that's a outgroup of the sponges, and the cnidarians are the radiata. And then we have the bilateria, who are pretty much everything else. So here's a diagram that shows all of them. If I got tongue-tied on that, it's because I'm tired, and I'm trying to get these presentations together for you. Here's our stem group down here, the peripherans, and the eumetazoans are branching from one another down here on the left-hand side. And then when you get into the eumetazoans, we would break out the cnidarians and put them part of the radiata, and then we'd have the bilateria for everything else on here. So if we go right down the list, the echinoderms very commonly have fivefold symmetry, but that fifth piece of that fivefold symmetry commonly it's very symmetrical, in other words, so you can divide 5 by 2, you get 2.5 on one side, 2.5 on the other. That's kind of what you do with the echinoderms. For the hemichordates, we're talking about things like graptolites. Graptolites have a sort of symmetry about them as well, and it's bilateral, and so you can divide them in half. In other words, there would be half of one piece of a graptolite on one side, one of their stolons, they call them. And so a stolon is the dwelling place for a colonial animal that would have lived on a stalk, essentially. I don't know how to describe them. They're extinct now anyway, but they're hemichordates is what most people assign them to. We belong to the group that's called the chordates, and so we have a notochord, and then we had a spinal column after that. So we're part of the chordates. And then eventually that will become things like the reptiles. Well, actually everything before that, even the fish, were chordates. And the other group, it's called the prostomia. And so that's the deuterostomia up here at the top. And that has to deal with how these things actually form in an embryonic stage. And so the deuterostomes have an interior part, and then the protostomes, prostomes, not so much, but these are the bryozoans. We talked about bryozoans briefly because bryozoans and brachiopods are like this, along with a group called the phauronids. All of those, in fact, have lophophores as a feeding structure, the bryozoans, the brachiopods, and the pharonid worms. And if you look at this is called the lophophore trichozoa, and so that would include the annelid worms and also mollusks for that matter. So even mollusks belong to that group. At the very bottom down here, the ecdysozoa, that's the arthropods, oncophorans. I'm not really familiar with them, but there's a type of worm at the bottom. It's called a priapulid worm, and so the priapulids, or the priapulidae, are also part of that group. And so you're going to see some fossil versions of those. So that's our major stem groups out of the animals. So animals can be broken into, are they sponges? Yes or no. Are they cnidarians? Yes or no. They belong to the eumetazoans then, and then we can break them into the deuterostomes, the protostomes, and the ecdysozoa. If we have a look at just the structure of these things, we're looking at actually, in fact, in this case, how do you compare sponges with cnidarians, the polypoid cnidarians? And they're quite different, in fact. That's a sponge on the left-hand side here. This is somebody's illustration that would indicate how these things actually fed. They would grow in a vase-like structure. They would have an area in the central that's called central cavity. At the very top is the opening. That's called the osculum. And if there are currents that go over the osculum, it draws the currents into that central cavity through these pore spaces. And so there are collar cells that surround those pore spaces, and then inside of the sponge are these sort of feeding sort of like cells. And so if a protozoan gets sucked into the sponge this way through the exterior and then siphoned out the top by the currents that are going through, they may get caught or they may not get caught. But if they do get caught, then they get caught and digested and incorporated then into these sort of cells that digest the food. They're looking for things that are floating in the water column, protists and things like that, maybe some radiolarians, maybe some dinoflagellates and things like that as well, or diatoms. On the right-hand side, you can actually see the polypoid version of a cnidarian. You can think about those as like sea anemones. Sea anemones have these arms, right? And those arms are responsible for trapping those same sort of protists, but in this case, it's not a passive feeding structure. It's actually active, and so they can grab onto things and channel it into that opening. And when they do that, they're able to digest things in this enteron, that central cavity. And so these things have a coelom inside of them, in fact. And so the coelom is what allows them to then digest the food and so forth. And so they also have specialized cells in the exterior on the tentacles that have stinging cells. And so the stinging cells are called nematocysts. You can see what they look like when they're not triggered quite, but when something comes along, touches it, it'll actually harpoon into things like a fish, for instance, right? So anemones can actually feed off of fish, except for clownfish that actually clean the anemone. So that's a polyp version of an cnidarian here. Here are the two different life habits of an cnidarian now. You can see a more complex polypoid form on the left-hand side, and it actually is incubating some medusoids in there and will expel them at some point. They actually do what we call asexual budding in order to reproduce, and so a polyp will produce medusoids. Medusoids actually mate and form the polyp, essentially, that will go back to the seafloor, root itself on the seafloor, and form the next phase of life for this chain of living organisms that makes up the cnidarians. And so cnidarians have these alternating lifestyles. At least some of them do. Some of them are medusoid only, and other ones are polypoid only, I think. So, but they produce sexually and asexually both. Sponges, as far as I know, only produce by budding. And so this is a little bit more complicated sort of lifestyle. And I'm pretty sure most of the other eumetazoa actually have at least some phase that can be an actual sexual reproduction as well. So, yeah, it's a little bit more complicated when you get into that, but it also allows for more rapid evolution when you do that as well, because you're combining the genes from two different organisms. And when you do that, you're actually rolling the dice with a little bit better odds, I guess, at producing something that may be successful. So here we have the different lifestyles for the cnidarians. If we go on to the next one, we're looking at the radiata versus the bilateria now. So these are both eumetazoans. And for the radiata, you're looking at Mawsonides spriggi over here. So spriggi is named after Reg Sprigg, right? Mawson was his teacher. In fact, Mawson was an Antarctic explorer. The stories of Mawson in the Antarctic are worth reading if you ever get a chance. So Mawson was an Australian Antarctic explorer. He was trapped at one time in snowstorms. They had dog teams at the time. He wound up having to eat some of his dogs in order to survive. Otherwise, he would have starved to death. And I think some of his team actually were lost. But he survived and made it back to Australia at some point. He became a professor. And then Reg Sprigg actually studied geology under Mawson. Mawson was a geologist as well. And so this is named for Mawson here. It looks like a jellyfish, and that's what most people interpret as. You would think the same thing about the right-hand side. That's Dickinsonia again, and we've already talked about Dickinsonia. And I may have said something like saying that sure looks like a medusoid, but in fact it has bilateral symmetry, so it's not. And in fact is part of the bilateria, so it has to be part of the stem group for the bilateria is what most people think now. So it and the radiata are pretty darn close. So, but most people say, yeah, it's probably a part of the bilateria. So that is Dickinsonia, part of the stem group for the bilateria here. Both of those were from the Ediacaran. We're going to get now into the base of the Cambrian. So that's where we're going with all of this sort of phylogenetic sort of talk that we're going through here. The trilobites were the impetus for basing the base of the Cambrian on the first trilobites that occurred there. Traditionally, that's what everybody would say. The first trilobites, that's the base of the Cambrian right there. And usually it was a trilobite called Phallotopsis. But in 2012, they changed their mind. This is a committee. It's called the International Committee on Stratigraphy. And there's a subcommission on Cambrian stratigraphy. And they placed the base of the Cambrian, in fact, at a trace fossil instead. So there are trace fossils that occur before the first trilobites. But in fact, those trace fossils are pretty complicated. And so they call it Trypicnus pedum is the name of that trace fossil. That's it in the middle photograph here. And here it is on the right-hand side in the beds that incorporate Trypicnus pedum here. And there's the first trilobite that I know of anyway. I think that is the first trilobite that was ever found in strata. at least in Laurentia, I think. Archaeaspis macropleuron. And so that is the first trilobite that's found. It's got spines all over the thing, and it's a pretty large trilobite, actually. It was Bruce Lieberman who did a paper on that back in 2009, I think it was, something like that, talking about what are these earliest trilobites, these phalotopsids. So phalotopsids would follow on after some of these earliest trace fossils. The trace fossils show up that show a complex behavior that's more complex than what we would expect out of most other things that are actually radiata. So that's probably an indication of bilateria and maybe even of worms, okay, is what they would suggest. And so that is Trichognis pedum. That's the base of the Cambrian. They actually define these for a certain group of rocks that are in Fountainhead, Fortune Head in Newfoundland. And it's the first occurrence datum. That's what that FAD stands for on this slide. So it's the FAD of Trichognis pedum. And it's not without controversy. So when people choose these sort of boundaries, they're human beings, and they may have a reason for doing it. Now, for one reason, this is some of the most beautiful rocks you can imagine anywhere on Earth when you're out on a peninsula. And on the tip of a peninsula that's, getting seawater splashed over it all the time, during storms and things like that. It's just aesthetically pleasing. And the other thing that makes it kind of cool is they're exposed right there on the sea floor. Oh, you can see it. Well, it's not on the sea floor, but it's on the rocks that are, 10 meters away from the sea crashing in over here. So it's a pretty cool sort of setting for where you would have the first occurrence here, but not everybody agrees. Once they define this, and they actually establish what they call a golden spike, okay, with these things, it's like, where is that place that we can compare to everywhere else on Earth? Where's that lowest Trichognis pedum in this succession? So you measure a succession, you describe it in great detail. Well, somebody came along after they'd already defined that golden spike, and they found it lower, like about 10 meters lower. And it's like, no. So they got the wrong spot. Okay, so if you go to it, they're going to have to redefine where the base of this trace fossil actually occurs, Trichognis pedum. So there's our beginnings of the Cambrian right there. The base of the Cambrian, you now know, is based on a trilobite. Well, it used to be based on trilobites, now it's based on a trace fossil. But some of the animals that show up then are unlike anything that we have on Earth today. And they have a weird name because there's not a good descriptor for them. They call them small shelly fossils. That's what they call them. They're small. Most of them, not all of them are small, but most of them are small. They have shells. Many of them, in fact, are phosphatic, but not all of them. Some of them are actually siliceous. Some of them are calcareous. But then they show up. And remember Cloudina? We talked about Cloudina at the very close of the Ediacaran. Cloudina shows up. We find those in actually some Cambrian age, some Ediacaran age rocks, latest Neoproterozoic age rocks in California, in fact. So Cloudina shows up in the California-Nevada border area. But there are some other sclerites that show up, and then they become very abundant all of a sudden in the base of the Cambrian. So at the very beginning of the Cambrian, there are some rocks that are referred to as Timonian, and they're actually from Siberia. So in Siberia, they have the Timonian stage, and then they have the Botomian stage, and then they have the Botomian. It's the Timonian, Atabanian, and then the Botomian stages. So there's like 3 stages of rocks that occur, and they find small shelly fossils throughout them. And so the small shelly fossils show up at the base of the Cambrian, along with all of those archaeocyathes, right? So there are reefs out here, and then in very special sort of settings, you get the small shelly fossils that show up. This is the Cambrian explosion. We're in it now. The definition of it is, it's the unparalleled emergence of organisms between 550 and roughly about 530 million years ago. roughly near the lower half of the Cambrian period. And so, in fact, we call it the Cambrian system when we talk about the rocks. The event is characterized by the first occurrence of almost every major phyla on Earth. Somewhere between 25 and 35, or 20 to 35 different phyla show up in the Cambrian over that 20 million year stretch of time between 550 and 530. Almost all present-day animals groups showed up, except for the bryozoans. The bryozoans are going to show up a little bit later in the Ordovician. Almost all became armored at that time as well, so it tells us a little bit about the life strategy. If you're a stromatolite, what you do to make a living is you photosynthesize. You have that chloroplast within you that's going to give you energy. It's going to give you the sugars, the energy that you're going to get from the sugars. It was figured out by predators. It's like, I don't have to photosynthesize. I could just eat something that's photosynthesizing and be able to take that into my system and get the energy that they have stored up in them. And there began predation. And it's still with us today. You and I eat plants and animals. Well, most of us eat animals, but not all of us do. Some vegans don't, right? So, and we have vegetarians, right, who get their protein from eating plants. And so that's where you have to get it from. We get protein, carbohydrates, all of these sort of nutritious things. We have to get that energy from somewhere. Otherwise, we would die. And these animals kind of figured that early on. And in order to avoid being eaten by other animals, you put the armor on, you put the chain link on, you put the helmet and the breastplate on, you put on the bulletproof vest, you get in the tank, you hope that a tank doesn't blow up. That's the sort of strategy that these animals were producing back then. And so there was actually an arms race that went along with it. And so if you want to survive, you had to evolve rapidly to get enough energy from other organisms to begin. Even if you're a trilobite, you're scavenging other organisms, right? Whether they're plants or animals living on the seafloor. Have to get your energy from somewhere, but then you also have to protect yourself so you don't become energy for somebody else, and so that you can do the things that you have to do, which is to metabolize and reproduce. And that's the gist of what that's the whole game for life, isn't it? It's like things will live and then live off of other things, right? And so that's what we do. It'd be nice someday we could actually photosynthesize and we'd all be green and very wrinkly, I'm sure. And eventually we'd be able to produce our own food. And then some life form, some predator from outer space would find us and start eating us. Okay, no, maybe not from outer space even. Maybe from right here on this planet Earth, they would come along, and the tigers and the lions and all sorts of things. We'll begin to find these plump little creatures that are green and walking around the planet. Okay, I've diverged a little bit from where my lecture was going to go here, but in fact, that's, it'd be nice to make that the perfect world, but in fact, we all have to eat and get our energy from other animals and plants, or plants. And so what do small shelly fossils look like? Well, this is kind of what they look like. These are phosphatic plates. Now, I know these quite intimately because I picked every one of these things myself and photographed them in the SEM when I was doing my master's thesis back in about 1987 or 88 or somewhere in there. And so this, in fact, is a series of small shelly fossils that are referred to as canardids. And canardids are actually a group that's now placed within the caminellas, I think it is. And so there are worms, essentially, that would have armored themselves with these phosphatic plates to protect themselves from being predated upon. And so there were certain arrangements that these things would form. I noticed that there were 3 different types, 3 different varieties of these things. And so I designated a Form A, Form B, and Form C, counted them up. How many did you actually recover from different samples? And so I built this reconstruction on the left-hand side over here. And since then, since I published that in 1990 with my advisor, Burt Rowell, so that was Evans and Rowell, 1990, in the Journal of Paleontology, In later versions of this, you can see the same type of animal, Camanella, crawling across next to an archaeocyath behind it there, and being able to scavenge off of the seafloor, likely, but a worm sort of shape with bilateral symmetry, obviously. So here is our reconstruction for some of these small shelly fossils. Much of that, in fact, is based off of one variety that occurs in the Burgess shale. The Burgess shale is Middle Cambrian in age, a little bit younger. The ones that I just showed you, the Daliaceae, that is, that were canardids. Daliaceae is the name of the genus. That Daliaceae, Odyssey, I think, was one of them that we described in this, and Bradycae was the other one. Those Daliaceae, in fact, were Bathomian in age. And so the third series within the Lower Cambrian, that's the age of those sort of fossils from Antarctica. They're from the Shackleton limestone. When we go to other places around the world, they find them in some of the earlier stages as well, especially in Siberia. So Siberia is noted for the small shelly fossils that occur there in the Lower Cambrian sort of strata. And so the Russians had been working on that for one or two generations before the rest of the world really kind of caught on to what these creatures were like. And so one of the classes that I took was reading Russian when I was in grad school. And it's like I recall 3 words now, maybe. But in fact, we have all of these small shelly fossils that likely are related to worms. It's Halkieria in the Burgess shale that resembles them, and Halkieria has a palm frond sort of shape, sclerite that it would have had, as opposed to these things which were kind of pointed and with ribs on them and things like that. But these things, most of them are likely related to the worms. But there's a lot of different kind of worms, right? So annelids are one variety of worm. The polychaetes are a variety of annelid, as I recall. And the priapulid worms are a little bit separate from that. The priapulid worms, in fact, are not segmented worms like the annelids are. So, but the arthropods, of course, had segments on them in the bilateria. There are many varieties of arthropods, obviously. The trilobites are just one variety. You're going to see more than you can imagine when I show you some photographs from the end of this presentation from the Burgess shale. So the chordates and the hemichordates show up. There are early chordates that are known as conodonts, and so conodonts show up because they had a primitive notochord that ran down their back, but they also had these grasping sort of like teeth, essentially. They were made out of calcium phosphate, just like your teeth are made. And so these things would have grabbed on to prey or been scavengers of some sort and ate dead things or on the sea floor. Now, a lot of people think that they looked a little bit like hagfish do today. So hagfish are these sort of scavenger sort of like fish that will actually, they're very primitive fish, but they can get into other fish and eat them from the inside out, essentially. But people think that conodonts had that similar shape to them, not the same life habitat perhaps. And so but they were probably still scavengers of some sort. We know that the fish were actually chordates, and the graptolites, of course, were hemichordates. We've already talked about the cnidarians. I won't go into them in detail. Echinoderms have that 5-fold symmetry that really you can take that 5th plate and break it in half, and you can see the bilateral symmetry out of 5 that way. Mollusks have a lot of different classes, and those classes would include the gastropods, the monoplacophora, the polyplacophora, and the bivalves. And so there were also gastropods, and then there were the rostroconchs. And the rostroconchs are very unusual. They look like a bivalve. They're kind of U-shaped, in fact. They don't actually have 2 valves, but it's one valve that's kind of U-shaped, and then they'll often have a kind of an area, a tube out the back of it, so it would be for their excurrent or incurrent siphon, in fact. So they're very likely related to bivalves in some sort, but in fact they were a branch that didn't make it anywhere, whereas the bivalves did. With the bivalves on here as well, so that's our mollusk classes to deal with here. And then of course we had the sponges and the other sorts of things, all right, as well. The outgroups, in fact, of the eumetazoans. Let's take a look at what they look like. There's a whole series of worms over here. I'm not a worm specialist by any means, but in fact over here on the left-hand side are a whole bunch of things that they're not the kind of earthworms that you would expect to find. Okay, so earthworms, in fact, are annelids, and those annelids, in fact, have a lot of organs in them. They're fairly sophisticated. And so many worms, in fact, were sophisticated, and they have these sort of like feeding structures very commonly. Phanerozoic worms would have a feeding structure, and so, as well as the priapulids and many of the other polychaete worms as well. So polychaete worms would include things. I think they're in the serpulids, actually. The serpulids are in the polychaetes, and so the serpulids, in fact, had a sort of feathery sort of structure that would stick out, and they would be able to filter feed some of the single-celled organisms that were the primary producers, in fact. That's a priapulid worm on the right-hand side here. The priapulid worm is very interesting. I got to share an office with a guy named Simon Conway Morris when I was in grad school. I also got to share an office with another guy named Stephen Bankson. And so between the two of them, they were two of the world's leading experts. I guess they're still leading experts, in fact, in the and also Lars Holmer. Lars is out here somewhere. Lars is a brachiopod specialist, but he's also worked with small shelly fossils as well. But Simon Conway Morris, Stephen Bankson, Lars Holmer, these are folks I got to rub shoulders with when I was in grad school at the University of Kansas. Quite an auspicious group to actually share the office with. Priapulid worms are actually named for the god Priapulus, or it's for the inflated male organ, if you will. I think that's where that comes from. But a priapulid worm looks a little bit like that. Some people refer to them as penis worms, I guess. But, I don't want to get this thing thrown off of YouTube, so let's just leave it at that. Here's another annelid worm. Okay, so annelid worms are segmented worms, whereas the priapulid worms are not. We have other types of worms like these. These are actually phanerozoid worms, and phanerozoid worms would have a lophophore that would in fact be able to like filter feed things out of the currents that would wash across the seafloor. Of course, all of these things are attached to the seafloor. Here's a brachiopod that is not attached, however. The one on the right-hand side, you can actually see the pedicle. That's the sort of thing that people would eat, perhaps in the Philippines. On the left-hand side, you can see it without the pedicle. And so that's a lingulid, actually, a lingulid brachiopod. It is made out of the same material that your teeth are made out of, at least the shell is. It's made out of, in this case, calcium phosphate, but also a little bit of chitin as well. So your teeth are not made out of chitin, but they have some calcium phosphate in them. So that is a lingulid. On the left-hand side, that's a lingulid. On the right-hand side, with the pedicle still attached, that's what they would use to draw themselves into a burrow of some sorts in order to protect themselves during low tide. During high tide, they would extend themselves out and be able to feed that way. It's one of the oldest plexuses of genera on the planet. So these things have been around since the Cambrian. They're still around today. They're kind of primitive, but they're not so primitive. I mean, they got kidneys. They got reproductive organs. They have sexual reproduction, even in the Cambrian. And they have these little tiny hairs that would stick out. And so you're going to get to see some of those in the Burgess shale. You can actually see the setae that would actually extend from the shell and allow these things to feel something go by, and then they could like suck it in, into where the lophophore would then be able to filter feed some of that material, perhaps. That's a brachiopod. So that is our brachiopod. So we go from the worms to the brachiopod here. Here's another brachiopod. This is a pterobrachioid brachiopod. You can actually see the lophophore. It's slightly gape along the commissure line here. Doesn't have the same shape as the two previous brachiopods that you saw, three previous brachiopods that you've seen. You've seen lingulids, you've seen the spirifer brachiopods, and you've seen another brachiopod that had a kind of a sulcus in it, or a fold in it. And so in this case, you can actually see that this one's more rounded or bulbous, if you will. Leaves more room for the lophophore to curl up inside of the shell here, but of course this thing makes its own currents essentially by pumping those little fine hair-like extensions on the lophophore to be able to generate currents that would draw things across it. What a successful, plan for a life. They've got this great shell that protects them from being predated upon. They also have the good fortune of not being tasty. Okay, so they don't taste very good. That's only one country on earth that eats them. Okay, so that's the Philippines. And so everywhere else is like, there's no meat on these things. Why would I want to eat one of those? And so that is a brachiopod. So that gets us back to some fossil organisms now. And we're talking about the Cambrian. I'm just showing you pictures of living things that were around back during Cambrian time. This was an actual Cambrian organism. It's not around today, in fact, though. But it had this sort of film preservation of it in the Burgess shale here. And that's a thing called Anomalocaris. Now, so Anomalocaris is pretty cool. We know pretty well that Anomalocaris was actually a predator. Anomalocaris had feeding appendages. It had like arms that would go out, and it actually had grasping sort of like claws at the end of those arms. And it also had a mouth that people misidentified as a separate fossil. But the fossil, when the thing broke apart, when it died, it would leave behind the mouth parts, and it would leave behind the carapace, in this case, in special preservational circumstances where you get a film. But in fact, that is Anomalocaris. Here's the reconstruction for Anomalocaris. Now that's Patoia over here on the right-hand side. That's actually the mouth of Anomalocaris. You can see it on the reconstruction on the left-hand side. You can see the feeding appendages out there at the front and the eyes off the side. And this thing would be able to swim around, and they're pretty sure that this thing ate trilobites, or at least pieces of trilobites anyway. There's a couple of other things that are pretty interesting in this artist's reconstruction as well. Okay, that's a trilobite up here at the top, swimming around with the big spines on it. Did they have spines like that? It didn't want to get eaten, so it had spines on it. That's right. And so what's the thing down here at the bottom? It has spines on it too, notice. As a protection against predation, probably. But it's called hallucigenia. It's named after a hallucination, okay? So whoever described this first, it's like, what kind of animal is this? You know, it's like they actually, when they did the reconstruction for this animal, they had it upside down. So the spines are actually a protection. And somebody had it like walking on peg legs, essentially, when it was first proposed as an animal. But so hallucigenia got a remodel back during the early 2000s, I would say, maybe in the 1990s even. Here's one of the feeding appendages for Anomalocaris. You can't see the grasping so much on here, but I think each one of those little spikes on there would be able to like grab a hold of something, perhaps maybe jam it into or wedge it in between the axial segments of a trilobite to be able to hang on long enough to be able to take a chomp out of it. So that is Anomalocaris right there. This is a trilobite. And so, yeah, man, they had some armor on them. So this I think this is actually a reconstruction. Well, obviously it's a reconstruction, but they have found some that were like this, where it takes people months to be able to actually prepare fossils like this. You have to very carefully extract all of the matrix around the fossil. I think this one was probably 3D printed here. But you see the spines that were there for protection, right? Against predation. Here's the more common trilobites that you would see. So I think that's Elrathia kingi on the left-hand side. And maybe on the right-hand side as well. I think Bathyriscus is another fossil that shows up at this time as well. But there are bite marks on the sides of these trilobites. One of the colleagues that I shared an office with in grad school, his name was Lauren Babcock. He's since gone on to write some textbooks and things like that. And he did an article in Science. It was Letter to Nature, I think it was, originally. And then I think he did a Science article later on, but he talked about predation and healed injuries, in fact. How do you know it was predation? Well, this thing actually survived the attack that left that scar. And then the scar kind of was left behind when this thing healed. It goes to show you that these things had an arms race going on between predators and prey. And of course, the eyes were one of the survival strategies for the trilobites. They were able to see all the way around them, in fact. And I think I mentioned also a camera in the previous lecture about this, but there's a camera that's now based on the eyes of trilobites to be able to detect a deep field of focus, a deep field with that sort of compound eyes that these things have, like a fly. There are some special circumstances in some places around the world, and this, I think, is from the Orson shale. Actually, it's the alum shale, and the Orson is a layer that has a whole series of siderite concretions, and those siderite concretions can be broken open. And inside of them, very commonly, are microfossils like this, and they found, in fact, some early ostracods in the Orson, I think it is. And so these ostracods are bivalved varieties of arthropods, and so that's an arthropod you see here. In fact, even many of the appendages, most of the appendages, the gills even, were shown to be preserved inside of many of these phosphatized ostracods in this case. And so really quite an amazing sort of preservation in this thing. And so people take an SEM to photograph these things in great detail. Really, truly astounding fossil preservation in this. So we know lots about Cambrian arthropods, not only from the Burgess shale, which is a special circumstance, special variety of soft-bodied preservation, but also you also get special circumstance for hard-body preservation and the permineralization of some of these, or the replacement of some of these organisms. So here's what an ostracode looks like. That's a living one, a modern one today. So ostracods, okay. Ostracods are preserved in the Orson, but they're also in the Ordovician of, let's say, the Great Basin. Some of them got to be about almost 2 centimeters long, centimeter and a half long, 15 millimeters long. Absolutely enormous for a bivalve kind of, that had a protective coating on the outside. It didn't have to rely on rolling up in order to protect itself from predators, in other words. And they're very abundant. And so ostracods like this were able to make the jump from marine water to fresh water. And in fact, in any pond in Missouri today, you're likely going to find ostracods like this. So yeah, pretty cool, very successful sort of body plan for an organism that's been around since that Cambrian explosion again, right? If the chordate showed up, then we have the fish that showed up. We have the hemichordate, all these different groups. They all show up then. And most of them you can trace back to what their lineage is today. So here's an arthropod. The crustaceans were around back then. Of course, this is a crab. That's Mollusca. Here we have this is actually into the mollusks now. This is a variety called the monoplacophorans. So the monoplacophorans had only one shell, mono, right? And so, but the monoplacophorans, they didn't find them till I think it was 1953 that they first found Vima, which is a monoplacophoran in the ocean trench that's off the west coast of South America. So that's Vima right here. But we suspected that monoplacopherans were around for a long time because we found their fossils in the Cambrian. And here, boom, there's one that's living today. So there's a monoplacophoran that's living today. So most of the ancient single-shelled organisms like that are referred to as monoplacopherans. We also see things like this in the Cambrian, polyplacopherans. So polyplacopherans meaning many valves or many plates on it. And in this case, this is one that's living, obviously, or it's living on a shell. That's actually from Jamaica here. And these things actually eat algae off of rocks and anything on the seafloor, any sort of hard surface. They'll go along and graze on that surface, eating the algae that grows there. They have a tendency actually to dish out the area where they're living, and so they'll actually abrade part of the rock that's there as well. So these things have plates on their back. They can articulate some, but man, if you ever try to pry one of them, you're going to kill it if you try to pry it off of a rock. They are so strong, and they're holding for dear life really onto that rock. And so they're trying to protect themselves from the wave agitation and from being predated upon. So they developed an apparatus for like clawing their way onto the rock and staying there. And so there are even like small plates around the perimeter on the mantle of a polyplacophoran, as you see it in this photo here. Other bivalves you can see here. Here's the one on the left-hand side. Those are all bivalves on the left-hand side. The gastropods on the right-hand side. You've already seen them preserved as steincairns, but here's some that are like Turritella, but they're high-spired gastropods. So high-spired gastropods are around with us today. The bivalves obviously are as well. Here's the The nautiloid is around today, mostly in the deeper oceans. They swim and or look something like an octopus, essentially with a shell, and so they're able to regulate the pressure in the shell and change the height in the water column that they're swimming and so forth. And they can jet out just like a squid or an octopus can. And so that is an example of a shelled cephalopod here. They got quite elaborate, in fact, back during the Mesozoic. Much of the Mesozoic is defined by the marine strata anyway, or defined by the cephalopod shells that occur there. And so some of them got long in the Paleozoic. Some of them were even straight cephalopods, so it made it difficult to like aim themselves because they're swimming in the opposite. They're pointing in the direction they're swimming, but that point is not where the body is. The body is at the opposite end out here, jutting out water to be able to swim with the tentacles and so forth. So that's a straight cephalopod down here on the right, on the bottom, on the left, on the bottom. On the right-hand side, you see all these coiled cephalopods, but some of them really got kind of elaborate J shapes. They had all sorts of weird and exotic sort of shapes in these sort of things. How big do they get? Some of them are like 2 or 3 meters across in the Jurassic. So I mean, like things that are almost as high as a ceiling. And then some of the straight cephalopods got up to like 10 feet long, which is just utterly astonishing that animals could be able to do that. So I've talked a lot about the Burgess shale already. You haven't seen very many animals. You've seen a couple of animals from that already. The Burgess shale is an example of a variety of fossil preservation that we call Lagerstätte. It's a German word. It really means exceptional preservation. And so Lagerstätte is singular, and Lagerstätten is the plural version of that. And so here's a whole list of Lagerstätten that belong to the Cambrian. Just in the Cambrian alone, we have all of these different separate and distinct records of exotic animals and exotic preservation in each one of these. I'm only going to show you the one. I'm going to show you the Burgess shale. But many of these things, like the Weeks Formation, has some soft-bodied animals in it. I had another colleague at the University of Kansas who studied trilobites from the Weeks. Lauren studied trilobites from the Wheeler Shale and from the Spence Shale as well. So Lauren Babcock. And then there's all these, you've seen the preservation in the Orston and the Alum Shale that's down here at the bottom, Kinnikela. And then, so what else is on here? Well, I'm not really familiar with Blackberry Hill or the Kali formation here in China, but I've heard of the Mao Shan shales, and those have uncalcified trilobites in them. So they're quite old, those things, in fact, but they're unusual sort of preservation. So if there were trilobites that had shells, there were trilobites that also didn't have shells, but you had to have special circumstances in order to preserve them. And so let's take a look at some of these things. So here is an example of what it looks like in an artist's rendering of what these animals from the Burgess shale would look like. Now, pay attention to this, I guess I would say. What you're looking at here are kind of muds that are off the edge of an escarpment. So the back of this drawing, in fact, is the escarpment. It's called the Stevens escarpment, I think, there. But the Burgess shale was deposited off the edge of that sort of reefal sort of like setting, and in the Middle Cambrian, in fact. And so it was people have argued, is it structural or is it actually something that was actually a growth off of the seafloor there? There was topography, there's no doubt, but these animals got buried. So you see things on here like Wowaxia that we were talking about, sclerite-bearing organisms. Well, that's the thing that you can see Patoia here on here as well on the left-hand side. So there's sponges on here. Wowaxia is the one that's number 24 on here, if you can read that. It's a little difficult for me to read. I need glasses, I know. There are things on here that people don't know what they are, but the priapulid worms are down here at the bottom living in the sediment. There are things like Opabina over here that has exotic sort of like attachment at the top to which the eyes would attach. So it's like a trilobite with a stalk out the front. and the eyes would be on either side of that stalk. There are things that are bivalved arthropods on here, like number 29 on here, but mostly sponges and things like that. So let's take a look at these things. You're going to look at some of the evidence for this early radiation of exotic Cambrian animals that filled these niches, in this case, in relatively deep water off the edge of an escarpment, so kind of like intermediate sort of like depths of ocean water. Brachiopods here, so that's a charophyte, I think, related to it. Something that looks like a crab over here on the right-hand side, that's the Burgessii, Bella. And then I'm going to go through these relatively fast. So here's another sort of bivalve, sort of arthropod-looking creature. And so you can see this is a Canidaspis perfecta here. For other sorts of organisms that were likely related to, well, here obviously is a trilobite on the right-hand side, that Morella splendens. And then some sort of soft tissue next to it preserved as a film. And I'm not exactly sure what that is. Helmetia, helminthes are very commonly worms, and so Helmetia is probably some sort of worm, but it was an armored worm, if anything here, and you can kind of see the unusual plates that are on the back of that sort of thing. Here's another arthropod. It's in the bilateria, obviously, and you can see a head region and a tail region on it, with spines on it as well for protection, I assume, and then maybe some like antennae that are off of the sides on the head regions up there. That's not a trilobite though, but that's Neroia compacta. Here's another one, Odaria alata. And so again, something that resembles both worms and perhaps even an arthropod here. So arthropods likely came out of the worms anyway. And so it's not surprising to see that. Here's some trilobites. Here's a trilobite on the left-hand side, and then a sort of shrimp-like creature on the right-hand side. So Olenoides is a shrimp, or excuse me, a trilobite that has many of the later appendages. You can actually see the legs sticking out from underneath of it there. On the right-hand side, a shrimp-like animal. Here's another shrimp-like animal, Waptia fielensis, and so another shrimp-like creature with antennae out the front. Here's one that was more segmented, if you will, Yohoia tenuis, and maybe more worm-like in this situation, but remember the worms also are bilateria. Here's one, Burgess Okita, Stigeria, and so Stigera. And so this thing had like brushes, like almost for its appendages off the side here, but certainly bilateral in symmetry. Here's one that's a little bit more difficult to interpret, I would say, but again, either worm or arthropod in its relations. Canadia spinosa, lots of spines on it. Here's that priapulid worm now. Here's the Otoya prolifica, right? So that is the shape of that worm with the head regions up there on the left-hand side, tail regions on the right-hand side. And again, something that would have crawled through the sediment here. Selkirkia columbia, I'm not exactly sure what the affinities are that people have associated with it. Iphelia globosa here it has a lot of stars on it here. Those things have been called chancelloria before. And so each one of those individuals would be chancelloria. And collectively, they're a chancellorid sort of animal, I guess you could say. But in fact, they were probably spicules on a sponge. And so here, probably attached to that sort of creature that was on the bottom, because sponges have to have a hard bottom to attach to, usually. Here's some brachiopod sorts of things. So that's Dictynina panella here. And you can actually see some of the setae that are sticking out of the shells there. And I'm not sure which one of these is Piranha mercata, but I assume it's the one that's at the top there. But here we have Picea. Pica is a little tiny rodent that lives in the high mountains of the Rockies. And so this is Picea gracilens. And so again, a worm sort of thing that's a lot like Amphinura, I think it is. Those are living today, the Amphinuras that are living today. And so it's like, what are they called? Flatworms, I think they are. And so very, very unusual sort of things. This one's more difficult. It looks like a tulip almost sort of shape to it. But I think people have referred this to some sort of polypoid sort of creature, but I'm not sure. I'm not sure. But yeah, Dinomischus isolatus. And so here's that Hallucigenia sparsa here that had the spikes and then also these other appendages. Probably walked along one side and the spikes were probably sticking up for protection. Here's that Opabina that I talked about with that one appendage, the eyes on stalks out here. So the eyes aren't actually on the extended stalk out there, but it has that sort of proboscis sticking out there. So Opabina regalis here. Here's Wilwaxia. Now, this is one of my favorite ones because this is what many of the Dahliaceae and the Camanella were. Their reconstructions are actually based on the idea that these sclerites would actually armor that creature. And so it makes you wonder, were they more linear and then got flattened essentially? And we know it's flattened somewhat, but these are more like palm shaped instead of having that sort of like rigid cone-like structure that was kind of serrated and ribbed, in fact. But Wilwaxia, another one of the Burgess animals here. So you see, the Burgess shale is loaded with all of these creatures that we really don't even know the affinity to with certainty. Most of them we can kind of guess what they are and say that there's certainly affinities with worms or arthropods or maybe even fish. I think the one that is the fish was actually the one that I said was maybe a flatworm. I think Pyke has actually been referred to as perhaps a fish even. So and the earliest fishes didn't have jaws. They didn't have much other than gills and a few muscles to help propel them through the water. But Wilwaxia, again, many of the sclerites that are bearing organisms from the lower Cambrian, from the early Cambrian, had that sort of armored appearance. And so, they would have had some sort of mantle that secreted that skeleton. So it was an arms race back then, as well as a massive evolutionary bump, if you will, out of the Ediacaran. So if the Ediacaran had these unusual sorts of animals, all of a sudden, by the time you get to the Cambrian, things are putting on skeletons. If they were around before, they evolved these skeletons relatively rapid in order to avoid predation. The predators were there. We know that as well. So there's this pressure for animals to develop quite rapidly. And so during Cambrian time, it's a major bump in evolution on Earth. And so did it happen all of a sudden? People argue about that a lot. Yeah, what's 20 million years to a geologist? We have to keep it all in perspective, right? Because the base of the Cambrian at 540 million years is really just about 11% of geologic time. And so we went through 90% of geologic time with hardly any change whatsoever, other than a few stromatolites, maybe a few protists and things like that. All of a sudden, we had the Ediacaran, some things start showing up with bilateria, and then also the radiata. And so you have these sort of primitive organisms that were around and left their traces behind. But then all of a sudden during Cambrian, boom, things hit. And it's just like this huge proliferation of animals at that time, between 550 and 530. So this big bloom happened right after glaciation. It happened after a big continental breakup. We had Rodinia. Some people also refer to it as Pannotia, but normally we call it Rodinia, roughly around 760, 750 million years. So relatively rapid. We had the evolution of the Ediacaran, and boom, right on its heels, the Cambrian explosion hits. Right on the heels of the Cambrian explosion, we're going to experience another event. And it's going to be a good one, actually, because it's important for evolution on this planet. It's going to be called the Great Ordovician Biodiversification Event. We're going to go to that next. Anyway, thanks for your help. Thanks for your attention. And this is plenty long enough for this presentation. I'm sorry it took so long, but it's really one of my passions. It's something I know a lot about because I've actually done some research on it. And I've done some on the Great Ordovician Biodiversification Event, but the presentation won't be quite as long as what this was. But anyway, this is kind of neat stuff, and it's the stuff about what life first got kick-started here. in the Cambrian. So anyway, thanks a lot. I'll talk to you soon.